Hello and welcome to the class on design of experiment. Let us try to understand some of the definitions before going into the details of the design of experiment. The objects of comparison which are applied for assessing their values are known as treatments. The treatments can be medicines, feeds, vaccines, surgical procedures, etc. The material on which the experiment is performed is known as the experimental material. In case of veterinary or animal sciences, cows, buffaloes, bulls, goats, or poultry can comprise the experimental material. The experimental material is divided into a number of groups to which the treatments are applied. These groups are known as experimental units. Most of the times we use the words trial and experiment synonymously. However, consider the experiment to be a larger entity which is formed by the combination of a number of trials. For example, in an experiment of tossing 4 coins, we may consider tossing each coin as a trial and therefore say that there are 4 trials in the given experiment. Any planned experiment which is involving human or animal subjects, which is designed to assess the effectiveness of one or more treatments or preventive measures, is known as a clinical trial. Biases in a clinical trial can be categorized as selection bias, observer bias, publication bias, allocation bias, and assessment bias. Design of experiment is nothing but the plan of proper allotment of the treatments to the available experimental units. A well-designed experiment has a well-defined method of statistical analysis of the data which is collected under the given experiment. The need of design of experiment is to detect certain specified differences between various treatments to get maximum information out of possible resources and to avoid the various biases. The basic principles of design of experiment include randomization, replication, and the local control. Randomization denotes unbiased allotment of treatments to the experimental units. Randomization ensures an equal chance for all the experimental units to be included in any particular treatment. The animals should be allotted such that all animals have the same chance of receiving any treatment. The allotment of one animal to a particular group has no influence on the allotment of any other animal. We should not know in advance the treatment that each animal is about to receive. There are various methods of randomization such as simple randomization, stratified randomization, restricted randomization, and group randomization. Simple randomization is the basic randomization procedure which does not involve any refinement or restrictions. Suppose we have two treatment groups, the test group and the control group. We can randomly allocate the experimental material to each group based on the random table method. In case of stratified randomization, the population is divided into different strata according to the categorization of the variables. For example, we can stratify the dogs as large, medium, or small breeds. Now, within each stratum, we randomly allocate the dogs to each of the treatment groups. Generally, a researcher aims to have approximately equal number of animals in various treatment groups. The restricted or blocked randomization helps allotment of equal number of animals to each group. In case of restricted randomizations, once we have allocated the required number of animals to one treatment group, we should allocate the remaining equal number of animals to the other treatment groups. In case of human medicine, an individual will be the experimental unit. However, in case of veterinary or animal sciences, group is the most appropriate experimental unit because food, drug and vaccines are often administered to a group of animals in a litter, pen, paddock or a tank. Now when the randomization procedure is applied to the group, it is termed as a group or cluster randomization. Next principle of design of experiment is the replication. Replication means repeating the number of measurements of the same type on each experimental unit. The number of times the effect of treatments is measured is referred to as replication. The replication can also be when a certain response is measured on several occasions on each experimental animal over a period of time. Repeated measurements are extremely important to obtain a proper estimate of error. However, increased awareness of restricting the number of experimental animals due to ethical and legal issues strongly encourages the use of only limited number of animals in a trial. The next principle of design of experiment is the local control. Here, the experimental units are deliberately separated into various groups. The blocks. The block acts as a local control. The animals within a block are more homogeneous, they are more uniform. The variability within each block is smaller than that between two different blocks. Incorporation of a careful grouping of the experimental units in a trial serves as a supplementary technique to reduce the variability in the comparison. Animals are allocated within each block to the various treatments randomly. Local control makes it possible to separate the variability in the results due to blocks from the variability due to the treatments. It allows a more precise estimate of the effect of the treatments. The principle of making use of greater homogeneity in a group of experimental units for reducing the experimental error is known as the local control. Thank you.